All right. I have to say, I'm really glad to be a part of the session, just personally from the perspective that I, our lab and my PhD work was on looking at or AH degradation and green infrastructure. And then we've also been doing some stuff with bioremediation and of thyroid compounds. So this is really fun. But today I'm going to be talking about some of our lab's work on transformation of neonicotinoid insecticides. And we'll kind of dig into what this is. So neonics have, or as they're most widely known, and people probably know about these from their impacts to beneficial pollinators, right? But in only relatively recent time, they've become the world's most widely used insecticides. And you can see here in the Midwest, we're in ground zero in they've been widely adopted for in agricultural applications, widely used in seed coatings. And so this imparts the most mass into the environment is due to these seed coatings. They're also widely used even in a lot of consumer products, including things like flea and tick medications for pets, as well as other things. So they started out just for specialty crops and became used all over the place and are used in a lot of other things. Chemically, there's a couple of really important things that I want you to know about them. First is that you'll note the KOW, right? These are very water soluble pesticides. Unlike a lot of legacy pollutant types of pesticides, these do not like to stick to sediments. And so we've seen them ubiquitously in surface waters in the Midwest because of the fact that they are very water soluble. The other thing is that neonics have what's called a pharmacophore, and which sounds a lot like pharmacy or pharmaceutical, right? What it does is it is some sort of chemical group, in this case, either a nitro or cyano highly electronegative group that goes in and confers selective toxicity by binding preferentially to insect acetylcholine receptors and are therefore repelled by a vertebrate or a mammalian, which is good. They're target specific pesticides, right? So they kill the pest more than they kill, other things. But the problem is that they can actually, if there's some sort of environmental transformation that affects that pharmacophore, it can actually go in and reverse that binding specificity and make them more toxic. So keep that in mind. That's an important point. And remember, because of this high tox or this high water solubility, these have been found all over the place. In 2017, we published the first paper on demonstrating that these are found in our drinking water. This, made quite a splash in the academic literature as well as the popular press and stuff like that. But honestly, we weren't really surprised, right? What is conventional drinking water treatment designed to do is to remove particles and pathogens. And for highly water soluble emerging organic contaminants, it doesn't work very well. So one of the things that we did in this and follow-up work was we looked at the University of Iowa and the Iowa City treatment plant. One of the things that we found was that the city of Iowa City actually uses a GAC filter for taste and odor. And this appeared to remove the neonics very well if we looked at just the finished and the source water and compared these. And the university didn't remove anything in comparison. We did follow-up work and we found similar results with the metabolites focusing just on the GAC filter. So this was really kind of curious because why does it work well to go in and for GAC to remove neonics, which are not hydrophobic contaminants, right? Usually we think of increasing KOW, increased removal via sorption, right? So we did a much more in-depth mechanistic follow-up study that was published here in 2020. And what we did here was we demonstrated that neonics actually soared pretty extensively to activated carbon. What's really interesting and what's curious about it is that as we go and we change that, remember the nitro group being highly electronegative, if we change that to some sort of urea or an amide, so it does nitroimidacloprid, that actually decreases the sorption. And so what we see is that just like the binding specificity to the receptors in the animals changes when you have this change in the pharmacophore, so does the binding specificity with the carbon. And so what we did was we dove into this a little bit more in detail using idealized carbon surfaces. We used functionalized and non-functionalized carbon nanotubes so that we could go in and change the surface chemistry by either adding something that's electronegative, like a carboxylic acid group or an amide group, so that we could go and we could see that how the sorption characteristics changed. What we could see was that electrostatic interactions are really some of the drivers of the sorption of neonics to activated carbon, which is different than the bulk hydrophobic interactions that most sorption is assumed to occur in for a lot of legacy contaminants. So we published this in 2020. And one of the things that we naturally wanted to know about is like, okay, there's only a very limited number of standards available for these anywhere, and particularly for these metabolites that are potentially out in the environment. One thing that we published in 2017 or 2019 was that if we took neonics or their metabolites, we could chlorinate them, like what would happen in drinking water. And we could find all these chlorinated disinfection byproducts as associated with the neonics. And so what we wanted to do is, well, how can we predict their fate, both on activated carbon for drinking water treatment, but also out in the environment, if we have different types of things that might show up on high res that we can discover by high res mass spec, either on the environment or by biotransformation or potentially by chlorination. But it's not practical to go through and synthesize these things one by one and try to try and create them. So what we did was we wanted to go in and probe how to predict them. So it's very common to be able to, in order to predict things in the environment, is to use the partitioning coefficient, right? We're trying to go in and see what is in different phases. And so a common way to do this is to use a polyparameter linear free energy relationship. This is a very common one used that's published for the so-called Abraham parameters. And for a lot of legacy contaminants, this works really, really well, but not so well for neonics. In this very classic paper, it overpredicts it by about 1 log unit for clothianidin when that is in their data set. And of course, that's the only one in their data set. So what we did was we took a computational chemistry approach to try and basically use a QSAR approach. And instead of going in and using their approach using the Abraham parameters, what we did was, again, what we were trying to do is match the KOW with a predicted KOW and look at what QSAR parameters really dominated the fit. So we could go in and try and look at this. What we could see is that if we look at what are the parameters that matter, it's the energy parameters that matter, as opposed to a lot of the things that are related more towards these bulk hydrophobic properties. And so what we did was we followed OECD guidelines for internal validation as well as external validation, so training and testing sets. And so next, what we wanted to do is we could see these role of the electrostatic properties again on here, because this is a surface electrostatic interaction. We predict that it's predominantly due to things like pi-pi bonding, pi-pi stacking that are related to the surface. For certain compounds, some of it is related to the interactions with the pharmacophore. As we probed more specifically for in that original study. But what we did was we created all these different sorption isotherms using, we fit them with the Freundlich isotherm, and then we went and used the carbon nanotubes, these idealized surfaces, and then went and plotted the Freundlich parameters, and then put them into our model, and then looked at the training and testing sets for these. What we then did was we extrapolated the log KF values for the CNTs, right? These idealized surfaces with GAC. So this is a much more realistic heterogeneous source. Then what we did was we went and looked at our experimental set, and these are experimental versus predicted for basically our training set, and we put these in, and then we had some different internal or testing validation and external testing validation. So these would be completely outside of our training and testing for validation. We recognize that this is a very small sample set because there's only so many of these compounds that are commercially available, and we didn't go in and synthesize any of these. So that was a really important thing that's kind of like a limitation and a take home and everything. We then went and we compared, okay, so how good of a model potentially is this, right? We take this computational chemistry, we go and we kind of do this regression analysis. There had been a paper that was recently published in 2021 using a machine learning approach, also using Freundlich isotherms to try to go in and predict sorption. What we found was that, similar to the Abraham parameters, that this was actually, their approach wasn't satisfactory and didn't fit well for neonicotinoids. Does that mean that their model's wrong? No, it doesn't. It just is that it's not appropriate for these types of more or electrostatically driven interactions that are present on there. So if we look at like our experimental, our predicted versus what we got a copy of their model that was published, we put it into the MATLAB and found it there. So one of the things that we can see here is that now we can go in and make predictions about what we can see for these different types of metabolites and these different types of transformation products from different factors. So let's just take imidacloprid for an example. Oops. What we can see is that it goes in and we can see that there is a decreasing sorption as you go in and lose that as you go in and lose that pharmacophore down there. We can see that increases sorption when we go in, which is consistent with the experimental results. We can go in and predict what would happen when it's chlorinated. We can go in, and these are some products that we don't have standards for, but we know are more insect toxic, and so we can make predictions about what these would be.
3 minutes.
We know that chlorination is predicted to increase the sorption to GAC, which is actually totally expected, right? So this is something that we see with all kinds of other types of contaminants as well. And so that's good that our model would be consistent with those fundamental predictions. We can also then go and, for example, see that transformation from the parent compound to the urea. And these are things that have only been found and reported in the literature by high-res mass spec, and we found these by high-res mass spec in some of our experimental work too during a hydrolysis experiment. So this was just published this year, and so it's actually available open access, so everybody can see it and use it and everything like that. And the conclusions from this work are really that the fate of target-specific pesticides, which are increasingly common, which have a very good intention, right? They want to go in and kill the pest without having as much collateral damage, can change greatly if you alter that pharmacophore. But we can go in and create models that are maybe don't have to have lots of experimental data, but if we combine the computational approach to it, we can go in and use that for predicting other things within that class, but more broadly. So this could be potentially used as an approach for things like PFAS, where you have different chain lengths, but you have fundamentally very similar chemistry, and they don't behave well with other classical models. So this work was funded by NSF and a whole team of folks, and I am happy to take any questions that you might have too. Leave up this conclusion slide. I think we have about 1 minute left.
Yep, about 1 minute. If anybody has a question, feel free.
We do have one in the chat. How confident are you in the experimental training data driving your new predictions?
Driving the new predictions that, yeah, I mean, these are, one of the limitations is obviously the sample size. These are because we are working with the carbon nanotubes, and these are idealized surfaces, they work incredibly well. They fit things very nicely in comparison to working with like more dirty environmental samples. Even the GAC is much more heterogeneous and soils, oh my goodness, right? But these are very, very heterogeneous or homogeneous and everything. So those work very well. What would be nice would be able to continue to expand this into larger things, but without more additional standards, we're limited by that somewhat.